This strategy is a powerful tool for comprehensively studying the global changes of plant phosphoproteomes in response to biotic or abiotic stresses. This technique allows in-depth phosphoproteomics analysis in an easy-to-use and high-throughput manner.
For IMAX stage tip preparation, use a 16-gauge blunt-ended needle to penetrate a propylene frit disc, and use a plunger to gently press the frit into the tip. Then, place an inverted nickel NTA spin column onto a 1.5 milliliter tube, and use the plunger to press the frit of nickel NTA beads gently into the tube. For phosphopeptide enrichment using an IMAX stage tip, add 400 microliters of loading buffer to the nickel NTA beads, and load the entire volume of bead solution into one IMAX stage tip. Pass the solution through the tip by centrifugation, and load 100 microliters of 50 millimolar EDTA to strip the nickel ions from the tip by centrifugation. Treat the tip with 100 microliters of loading buffer by centrifugation. Next, treat the tip with 100 microliters of 50 millimolar ferric chloride in 6% acetic acid by centrifugation.